Welcome back. Today, we are diving deep into human excretion. Metabolic activities produce various toxic nitrogenous wastes. Animals classify into three distinct groups based on how they eliminate these toxins. Ammonotelic animals like bony fishes excrete highly toxic ammonia, requiring massive amounts of water. Uricotelic birds and reptiles excrete uric acid as a paste, conserving the maximum amount of water. Ureotilism mammals, including humans, convert ammonia into less toxic urea inside the liver via the ornithine cycle. Today, we focus entirely on how the human body processes and excretes this urea. Macroscopic anatomy of the human urinary system. Human kidneys are reddish-brown, bean-shaped structures located retroperitoneally between the levels of the last thoracic T12 and third lumbar L3 vertebrae. Note for your exams, cortical nephrons have short loops of Henle extending minimally into the medulla. Juxtamedullary nephrons have very long loops dipping deep into the medulla, which are crucial for concentrating urine. The three steps of urine formation. Urine formation involves three distinct physiological processes. 1. Ultrafiltration. Blood flows under high pressure through the glomerulus. It is filtered across three layers: the glomerular endothelium, the basement membrane, and the epithelial cells of Bowman's capsule called podocytes. Podocytes leave intricate spaces called filtration slits. The resulting glomerular filtration rate, GFR, is about 125 ml/min. 2. Selective reabsorption. Out of 180 liters of daily filtrate, 99% is reabsorbed. The PCT reabsorbs nearly all essential nutrients, 70 to 80% of electrolytes, and water via active and passive transport. The descending limb of Henle's loop is permeable to water, but impermeable to electrolytes, while the ascending limb is impermeable to water, but allows active or passive transport of electrolytes. 3. Tubular secretion cells in the PCT and DCT actively secrete ions like H+, K+, and ammonia into the filtrate to maintain the body's ionic and acid-base balance. Mechanism of concentration, countercurrent mechanism. To prevent dehydration, humans must excrete concentrated urine. This is achieved via the countercurrent mechanism operating between the loop of Henle and the surrounding capillary loop. The vasa recta. Eat less than their big wit less than release than least than least less than least less than glog. It forms a countercurrent. Similarly, blood flows in a countercurrent pattern through the vasa recta. NaCl transported out of the ascending limb of Henle is exchanged into the descending limb of the vasa recta. This setup maintains an increasing osmotic gradient in the medullary interstitium, ranging from 300 mosmol/L in the cortex up to 1200 mosmol/L in the inner medulla. This high gradient draws water out of the collecting duct effortlessly, concentrating your urine up to 4 times its initial concentration. Hormonal regulation and micturition. Regulation is handled elegantly by 3 main feedback loops, less than ADH vasopressin, released by the hypothalamus when osmoreceptors detect water loss. It makes the DCT and collecting duct highly permeable to water, increasing water reabsorption. Bra's pathway. A drop in GFR stimulates the juxtaglomerular apparatus JGA to release renin. Renin converts angiotensinogen to angiotensin I, then to angiotensin II. This potent vasoconstrictor triggers the adrenal cortex to release aldosterone, reclaiming Na+ and water. Conclusion and exam tips. Make sure to study disorders like uremia, high urea in blood, renal calculi, kidney stones made of crystallized oxalates, and glomerulonephritis, inflammation of the glomeruli, for your upcoming exams. Drop your questions in the comments section below. Smash that subscribe button for more high-yield biology lectures, and good luck with your preparation. Thanks for watching.